Okay, so good morning, good afternoon, everyone. So today we have two famous guests, Carson Stringer and Jingjing Zhang. They will present this publication that I have on the screen. Farzaneh and I felt there was a so strong overlap with some of the things we've been talking the last few weeks that we were really excited to invite Carson and Jingjing to present so that we could potentially create bridges or discuss potential implications with all the work we're doing. I want to just take a few minutes to give a bit of context for folks that are joining in. So this effort today is part of a community project. We call it Open Scope Community Project. And so this is documented on this website here. You can go. There is a review that we did together with the community with more than 50 co-authors that went through key mechanisms of predictive processing to sort of design an experiment with the aim of designing an experiment to help move the field forward. And so that experiment is now in this publication as well as on the website, and we have started the experiment at the Allen Institute. So this is going to be released probably in the winter and across the spring, the data set, and for everyone to look at. So that's why I'm excited, and I think some of those experiments have interesting overlap with what Carson and Jingjing will present. So I'm excited to see what they have to show us. And then in addition, because we do this as a community project, our meeting today is going to be recorded. We plan to publish this on YouTube for everyone to see because some people are not on our time zones. So if anything occurred during the meeting that you feel should be edited out, please feel free to reach out and before the end of the day, and then we'll do that. That being said, Carson and Jingjing, welcome.
Thanks. You want me to share my screen?
Yes, please.
I think you might have to stop sharing.
I think you can override it, but I'll do it.
Okay, hold on. Let's hope for the best. I don't usually use Teams, and I'm on. Oh, here, I figured it out. Okay. Can you see that?
Yes, great slide.
Okay, great, thanks. All right, so I'm going to be presenting our preprint, and so I'm going to be telling you about Jingjing's work, and she's going to go in more detail in the paper, and I'm also going to preface it by telling you a little bit about Lin Zhang's work in the lab, because it kind of builds the framework for why we're thinking about these unsupervised learning rules. And we're lucky to get a lot of support from a lot of people at Janelia with our experiments. So in the lab, we do large-scale recordings using a 2-photon random access microscope, which it looks like you also have in the OpenScope project. So we are able to record from primary visual cortex and higher order visual areas simultaneously. We use dual plane multiplexing, so splitting the laser into two separate depths, so that we can capture 2 depths simultaneously, and we get around 50,000 neurons at a rate of 3 hertz. And this is in a transgenic animal expressing GChem6. More recently, we have started using these riboviruses from Sver Grodem and his colleagues, which enables, so basically because these are ribo-targeted, the expression is limited to the soma. And we've been getting really nice recordings with these injections. And so that's some of the data in the paper here today. And here are some example traces from those neurons and the segmentation from SweetTP. So we, in the lab, we're interested in this question of supervised versus unsupervised learning, as I think many of you are interested in this unsupervised learning question. So just to give you a context for supervised versus unsupervised learning, you can think of it in the context of machine learning first. So we have a neural network that gets its input images and outputs a label like cat or dog. You can train it in a supervised way, which I think many of you are familiar with. You have cats versus dogs, where each image that you put into the network is given this label while you're training. or you can have unsupervised learning, like a lot of this predictive processing that you're familiar with. In this case, the network gets images without labels, and the network has to learn basically from these non-labeled images. And so for each image, for example, in this example, instance recognition task, you could have different crops from the same image, and those need to be embedded similarly for this unsupervised learning problem. And then you push crops that are from different images apart in some of these example unsupervised learning algorithms. And so in the context of the brain, there's not that many unsupervised learning experiments. There are a lot of supervised experiments, right? We do a lot of experiments. Can you see my mouse, Jerome? Cool. So there's a lot of experiments, right, where people train mice to lick in one corridor and not lick in another corridor, the IBL task, monkey tasks, and so on. But there are far fewer experiments with unsupervised learning. There's some really nice studies from Nuo Li and Jim DiCarlo with some unsupervised learning. But most of the work in unsupervised learning in neuroscience has been on the theory side. And so one of the questions we wanted to answer in the lab is if we can test supervised versus unsupervised learning in mice. And you can see here one of these prominent theories is predictive coding, as you are all familiar with. So in the lab, what Lin did is he trained mice. So for the supervised mice, the task mice, he trained mice to discriminate leaf and circle corridors. So in each corridor, a sound cue is played at a random position. And in the leaf corridor, the sound cue is followed by a reward. This is what the task looks like. So in the mouse is in the circle corridor. It's not licking, stopping to lick because this is a trained mouse. It knows that that's not rewarding. In the leaf corridor, you'll see the mouse will, there it stopped to lick and collect the reward because this is the rewarding corridor. And so before learning, the mouse licks pretty equally in both corridors, but then after about 2 weeks, the mice quickly learn that the leaf corridor is rewarding and not the circle corridor, and so they end up licking in that corridor. So the mice are able to learn to do this texture discrimination task. And then one thing we wanted to know was how different are the representations that of the task in the task mice versus the visual representations in unsupervised mice. So this is a second cohort that Lin had that basically ran through these same corridors a similar amount of times with random sound cues, but they were never rewarded. So that's what we call our unsupervised mice. And so to discriminate, to find these kind of visually selective neurons, we use a D' index, basically the D' between the leaf corridor and the circle corridor. And that's how we find selective neurons. And so you can see for leaf selective neurons, you can see these neurons have tuning curves. They happen often, they seem to tile the corridor. They have specific positions that they're selective to. And then we're looking at their activity on the circle corridors. You can see they're not active there because we selected them that way. These are test trials. And then the same thing on the circle selective neurons, that we can see neurons that are selective to lots of different positions in these circle selective neurons and not active on the leaf trials. So we wanted to know where these selective neurons were in the brain. Oh, and feel free to stop me at any time if people have questions as well. Okay, so basically we wanted to look at where these neurons are before versus after learning. And here's our brain map here. Here's V1. Here are medial higher order visual areas, anterior and lateral, using the Allen Institute labeling. All right, so for neurons with a D' greater than 0.3, this is the absolute value. So we're taking leaf and circle selective neurons put together. If we look before learning, there are a lot of selective neurons in primary visual cortex. Then if we look after learning, there's actually a lot of selective neurons in these higher order medial areas, higher order visual medial areas, and also retrosplenial cortex. And what was surprising to us was actually we found a similar result in these unsupervised mice. So after learning, there was a big increase in the number of selective neurons in these higher order medial visual areas. Even in mice, these mice never got rewards. All they did was run through these corridors. So what this suggests is that visual experience alone is sufficient to drive a lot of plasticity, we think, in visual cortex, and in particular in these higher order medial visual areas. And kind of the last part of this story, so the paper has a lot more details on like Lynn does some really cool experiments with swapping parts of the corridor, showing different leaves and looking at generalization. But I'll just tell you the last part of this, that we do think this unsupervised learning is useful for tasks, which could be useful also for people thinking when you're thinking about predictive coding. And so we asked this question: does unsupervised pre-training, having this visual experience, improve your supervised learning experience? And so what Scott did in collaboration with Lynn is he had mice run through these virtual reality corridors for this. All right, so they got acclimatized first, and then they run through these corridors for 10 days without reward, and then they start the supervised learning. And each mouse had a different pair of stimuli from these 4 different stimuli that we showed. We compared these to no pre-training mice, so mice that were acclimatized and then only, and then went through the virtual reality corridor with supervised learning. And so this is, Scott and Lynn simplified the task. So basically they put a reward zone only in a specific place. So if the mouse licks anywhere in the reward zone, it gets a reward, rather than this sound cue and random reward. That makes it a little harder for the mice to learn. So this is like a simplified version of that. And then in the non-rewarded corridor, the mouse doesn't get a reward anywhere. And so on the first day of training, this is the passive reward day, the mouse always gets a reward in the reward zone. And then on the active reward days, the mouse has to lick to get a reward in the reward zone. And so if we look at a mouse that has gone through this pre-training for 10 days, on this is its first session with active rewards, we see that the mice quickly suppress their licking in the non-rewarded corridor across trials. But then they sustain their licking in the rewarded corridor, basically suggesting they can learn this in pretty much 1 day. After this passive reward day, they're pretty much experts almost. By day 4, they learn that the reward is in a specific region, is delivered in a specific region if they lick in that region, and so they constrain their licking to that specific part of the corridor as well. So we compare this. So basically, these are the curves across all mice with the VR pre-training of their lick response to the rewarded versus the non-rewarded. We can compare that to the no pre-training mice. So I will note, like, these VR tasks are very fast for the mice to learn with this fixed reward zone. You can see the mice are actually learning this in about 4 days. So it's still fast to learn, but you can see the performance is much worse on the first few days. So there is this improvement in performance from having the VR pre-training. And so we can quantify that by looking at the difference in lick response rate across days. And so on the passive, just starting on the active reward day, we can focus there. We can see this difference in lick response rate is big for the mice with this unsupervised pre-training, compared to the no pre-training mice, especially on the first and second day. And then these other mice catch up, because I think I don't have to tell people here, mice are smart and can use vision to guide their decisions. Yeah, so that's pretty much all I'm going to tell you about Lynn's story, just to kind of give you an overview before we go into Ching Ching's story. I'll give you an intro to that too, but does anyone have any questions about this stuff?
Yeah, I have a question for your one of the previous slides.
Yeah.
You show the effect of passively viewing on the distributed activity here, and I was curious if you tried a mouse that just watch a pre-recorded sequence and the wheel is not coupled to the screen, because we've seen here, we started to do this experiments and the mouse just completely change their posture, their behavior when the coupling comes on, and so I'm just curious if you've done this test.
That's a really good question. So Lynn has done a bit of that as well, and we don't see as big. It's been mixed. In some mice, we see some changes, and some mice we don't. So I think the closed loop is important to this aspect. And sorry, I didn't mention that. This is always in closed loop. So even in the unsupervised mice, it's always linked to their experience. So we think that is an extra aspect of engagement. Like you're saying, maybe the posture changes. I think the mice are more engaged in the task. So we do think that is important here as well, although we haven't rigorously quantified it.
Okay, makes sense. Any question in the audience?
And you'll see with Qingqing's VR as well, we also do that in closed loop. And so that could also be helpful for these predictive coding experiments as well. Okay, cool. Are there any other questions people have? All right, cool. Okay, so I'll give you an intro to predictive coding, which can be very short. I think you all don't need that, but basically, okay, so we think there is this substrate of, that there is unsupervised learning in the brain. We see these large changes just through visual exposure, and so we've been testing predictive coding in mice as one of these prominent theories of how unsupervised learning might work in the brain. And so what is predictive coding? So this is a nice illustration that Qingqing came up with. That basically you have this mouse walking through a forest. It walks through this forest many times, taking the same route, maybe from its home to some food source, and it sees the same sequence of trees while it's walking. And so, but maybe one day it's walking at this same path and it sees this Christmas tree. And so the mouse might be surprised, but it could be for two potential reasons. So it could be surprised because it was predicting into the future that after I see this tree, I'm always going to see this specific tree, and it's doing this temporal prediction. And then I see a Christmas tree, and this is not the expected stimulus, and so I get a predictive coding error. Alternatively, the mouse might be surprised because this is a novel input. So basically, the mouse has never seen a Christmas tree before. So it could be the case that the fact that this is a novel input that the mouse has never seen before is driving this, could be driving a surprise response or a predictive coding error. And so that would be falling under the framework of spatial predictive coding. And so often these two things are quite intermixed in the literature when people do experiments and talk about them. And so we really wanted to disentangle these things, and Qingqing did that in her experiments here. And so I'll go through these two types briefly. So for spatial predictive coding, you have the Rao and Ballard paper here, where you expect a lower response. For example, if you have like a long bar versus a short bar where there's like an error where the bar didn't complete outside of the receptive field of the neuron. Temporal predictive coding has been what a lot of people have been studying in the field. For example, the Gabornik and Bear study, where they show this sequence of ABCD, ABCD, and then look at responses when they hold out one of the stimuli, for example, and they see a change in the LFP. There's also been experiments from Pfizer et al. From Georg Keller's lab, where they do this sequence of BA, and then they omit A, and they see this big omission response, which could be indicative of temporal coding, although we have an alternative hypothesis in our preprint. So there are machine learning frameworks that could underlie these, that relate to these two concepts. So for example, in machine learning, people train these things called masked autoencoders to do this sort of spatial prediction. So as a way to do unsupervised learning. So basically they mask out tokens that they input to the network. So they divide the image into these little patches, which are tokens. And then the network is trained to predict the masked out tokens here. And that's the target for the network. Other unsupervised learning rules that networks use are temporal. For example, GPT uses next word prediction, suggesting that this could be a very useful framework for learning because GPT is quite a powerful model. So yeah, we kind of wanted to distinguish between these two. And so I'll let Qingqing, are you good to take over?
Yes, thank you.
Awesome. Okay, I'll stop sharing.
Okay. Yeah, let me share my screen. Okay, thank you, Carson, for the introduction. And yeah, if my nose starts bleed again, I'll just turn off the camera. We use the two types of temporal sequence to try to study this problem. One is with virtual reality. Another set of experiments is were done with static images, as you will see later. So this is the linear corridor we used. We have this ABCD sequence, so they are natural images. Each image actually is composed of two small images. They are exactly the same. So this A has two small A's. The reason to use those duplicates is because later we wanted to manipulate the duration of each image. So we can add more those small images or remove one of them. By doing that, we can introduce new temporal or new temporal kind of duration or interrupt the learned previous temporal sequence, but without introducing new input to the receptive field. So the mouse is running through this linear corridor for usually 5 days. This is how the response looks like after. Let me minimize this. This is how the response looks like after this training process. We sorted the cells based on their peak response in this linear corridor. Those are from test trials in V1. Those you can see those cells, they prefer the image A, and those cells they prefer the image B. They have maximum response to during those images, and also there are some cells they have maximum response in the gray, in the like random dots on top of the gray screen. Yeah, basically those cells. Yeah. Any questions?
It's a clarification. I think terms are important. The images here, they are positioned in space or in time? Because there's a navigation component in this task.
Yes, I think it's a kind of, if you think of position as the hippocampus literature, like a position means like real spatial position.
That's
It is
right.
coupled. Yeah, it is coupled. The space and the time, they are coupled. They cannot be separated. When we call like spatial predictive coding, actually we mean to the 2D input to the receptive field of every single cell. So it's a 2D image. Then that's the meaning, like spatial in this case. Yeah. So here, yeah, like you said, we would think if we use the hippocampus literature, like position or space, and also this time, it cannot be separated.
By couple. Yeah, no, that's fine. Just want to make sure we understand.
We
Yeah,
have
good.
a fixed speed, right? Changing that the VR goes at. So if the mouse is running above a certain speed, this is always it's always going to move at the same speed. So if the mouse runs through the entire corridor, then time and space are exactly locked. If the mouse stops or something, then we are interpolating to the positions from the time when it's at that position.
Yeah. Oh.
It's remarkable to me. So, sorry to interrupt again.
Yeah,
it's
sure.
remarkable. It's remarkable to me how uniform the number of cells that respond to each of the images and the inter-image area is. Like how sort of straight of a diagonal this line is. Is this a function of having trained the animals? Is this is only after training that this emerges? Is this like just how the brain is wired, or maybe it's just you've sampled enough neurons that this is what you expect? Can you comment on that?
My personal feeling is because I tried to use different image sets, and it's sometimes, for example, you might see this B group cells, they might be slightly less than the C cells, but roughly they like each of them like kind of occupied a significant portion of the whole population. I think one reason could be this: they are natural images. They are very rich in different features, so they can activate significant amount of cells. comparing to greetings. Yeah, I'm not sure if Carson has a different thoughts about that.
Can I ask one very explicit question? Does this distribution become more uniform with training?
Yeah, that's a good question. We do a baseline recording for every mouse, like before going through this training process. Like during this baseline recording, we can also sort the neurons in the same way as we did here. And it just they still like, because they are primary visual cortex and the neurons are still pretty selective. Usually the pattern will be slightly messier than this, and still similar amount of cells occupy the like a chunk in like here, and it just overall it's slightly messier than this, and overall the baseline firing rate is higher than this. I think it's they from the literature also they show that with training or this kind of unsupervised training over time, the neurons tend to be sparser than at the very beginning, and the baseline firing rate would be reduced over training.
Thanks.
Sure. So yeah, this graph is from test trials. We recommend to always use test trials. After training, for 5 days, in the next few days, we did a bunch of tests. We usually manipulate the image on the third position, so it's the C position. We either change the image to a normal image, so there are two small n's, or we change it to D image, exactly the same as the second D, or we remove half of the C, or we change it to a complete gray screen. Yeah, we'll see how the response looks like. When we change the image to a normal image, this is how the response looks like. Visually, it seems like there's a massive population level like activation to this novel image. We use the BD cells as examples to quantify this effect. The BD cells they have peak response to either B image or D image, and they are close to the C image. So you can see those cells, they originally their response to the C image is this big. When it's a no image, its response is this big. It's this dashed line. So statistically, neurons from V1, medial, lateral, anterior regions increased their response. And behaviorally, the mouse would slow down the running speed when they saw a novel image, and this is statistically significant across mice. We also changed the image to the D image, so the sequence became ABDD in this case. This is how the response looks like. Visually, we can see the response to these two Ds are very similar. Here when we do the statistics, actually we compare the D image, the first D from the ABDD sequence, with the original D from the ABCD sequence. So actually we didn't include this D for the comparison, just in case, because this D is following this D, maybe the mouse got lost and didn't know how to respond. So we compare this D with the original D from ABCD. You can see the neurons, like the D, for example, the D neurons, the response to the first D, and the D neurons originally responds to the very last D in the ABCD sequence. They are pretty similar visually. And when we do the statistics, there's no difference between those two Ds, and the mouse didn't slow down to check out the image either. We also, yeah.
Oh, sorry to interrupt. I have a question related to a connection with another study from Sonia Offer Laboratory, where I think they did something similar. And I talked with them. They said they would see error response like those only if rewards were involved in the task. That's what I recollect. And so I'm curious what could be the differences here. One thing that I'm thinking about is how far apart are your images from one another in space? Do you know that number?
You mean after seeing one image, how long it takes to see the next image? I think
Yes.
it takes about 3 or I think the in total sequence is about 15 seconds, and those random dots actually is slightly longer than the image. So I just I did try to make sure that when the mouse is in those random dots area, when the mouse is running, he couldn't see the previous image and he couldn't see the next image
I see.
either.
So
So yeah, there's like I think it's a 2 or 3 seconds period the mouse is in this.
So it's probably much closer than in Sonia Offer's lab study, right? I think it was 30 meters apart.
Yeah, I think that I remember their paradigm, the mouse take a pretty long to finish one trial, and they didn't have like hundreds of trials in one day. I think it's like I remember the trial number is much smaller than here, and the length is pretty long for one trial. And also they gave reward at the end of the corridor. Actually, the mouse didn't really need to do a task to remember the sequence. The mouse will get a reward automatically. Honestly, I don't know how.
They did say that without the reward, and as I understood it, there were no response like these in their and I think it's probably because the corridor is so long that you have to boost attention to see those
to engage.
Yeah.
And yeah, that makes sense. Thank you. Yeah.
Thank you.
We also blocked the treadmill, so in this case the mouse is passively watching the VR like a progress in front of him, and in this case when we have the novel image, this massive response was still there. Similar as in this case. We didn't really try to study the sensory motor mismatch error signal to see how this signal looks like, because we didn't accelerate or decelerate the VR to decouple with the mouse running like suddenly. Here we just completely block the treadmill. The point is we try to prove that the mismatch response we observed here does not rely on sensory like visual motor coupling. Basically the conclusion is the neuron behavior surprise response rely on novel image, not a novel sequence. Next we remove one small C, so this big image became half. Previously you might remember the response to every image is a duplicate. Like if you remove one C, that's only like one half of the response. And we quantify the response to the offset of this like one single C, and also compare the response with the response with the response after 2 Cs, and because those square when the line with like inside this square window is the after one single C, and here is after 2 Cs. We compare the response, and we statistically there's no difference, and visually we didn't see any massive response here, and behaviorally the mouse didn't slow down either. We next changed this C image to a complete gray screen, and this is more similar to the Keller paper, like Carson mentioned at the very beginning. Visually, we can see here that when the mouse entered this gray screen region, on the edges, the moment the mouse entered this region and the moment the mouse is leaving this region, there are some kind of messy response across many neurons here. We can see here on those edges, especially for the J cell are the cells that respond more to the gray screen with random dots rather than the images. We can see those cells they respond more comparing to when there's a natural image C. It's this dashed line comparing to the solid line. And statistically, it's a significant increase for those cells. Comparing also the behaviorally, the mouse would slow down to this gray image, to this completely gray screen, similar as the normal image. Yeah, considering for those naive mice, before the mice seeing ABCD extensively, we can see that those BD cells and also the J cells, actually they respond more when there's a C natural image. You can see here those solid lines, they are from ABCD trials. Those solid lines, actually they are taller than those dashed lines. But after training, we will have the gray screen, the dashed line is taller than the solid line. For the BD cells, maybe you can see it's pretty close. But here, the solid line is taller than the dashed line, which means that originally they respond more to natural image. After training, they respond more to gray screen. Yeah, originally it's comparing to gray screen, it's the response is higher. Now it's lower, and the mouse would slow down after training, but not before training. Yeah. So in summary. We observe a massive population increase response when there's a novel image and when it's a complete gray screen, and the mouse will also slow down to check out those images, but not when we shuffle the temporal order or reduce the duration of an image to half. Yeah, that's all about the VR experiment. Yeah, we also did one set of static image experiments. Yeah, it's kind of pretty close to what the people did in the literature, except for they are using grating stimuli. Here we use the natural images. They form this ABCG sequence. The duration of those images are 2 seconds, 1, 3, and 1 second. The mouse still needed to run, so the time of the duration will be counted. If the mouse is still and does not move on the treadmill, he would stuck with one image. After we still let the mouse watch this sequence for 5 days. After this training process, we did manipulations. We either changed the second image to a null image or changed it to gray screen. Yeah, this is how the event response looks like in test trials after training. Yeah, there are A, B, C, G cells. Still, the different cells, they prefer different images in this sequence, as similar as the VR experiment. This is how the response looks like when we included a novel stimulus. Yeah, I see. Yeah, might see that a big response to this novel image, and it's, I think here, and it's statistically significant. And also when we change it to gray screen, there's also a massive response here. Yeah, you can see the maybe even after this novel image, in the next image, this response is also taller, like in here. And after the gray screen, the next image, the response is also bigger. If you comparing to the response to the standard ABCG sequence. For the behaviorally, the mouse, if you look at this curve, it seems the mouse also slow down. It just I use the 4 mice in this case, like a one mouse didn't. So statistically, it's not significant. Yeah, but on average, it seems like the mouse tended to slow down in this novel image case. And when there's a gray screen, seems like behavior is not very dramatic. To really test the hypothesis, either the mouse is doing temporal or spatial prediction, especially for like a previous you saw those massive response when there's a gray screen. So to separate those two hypotheses, we designed a new experiment. Yeah, first we want to point out that when there's a gray screen, there's a massive response, we should, beside the mice might use a temporal prediction. It could also be, we should realize, and it could also be the input to the receptive field is different. You can see if one cell's receptive field is in here, when we change to a complete gray screen, the input to the receptive field is novel. The cell has never experienced before. And for the temporal sequence, for the static sequence, we should consider the spatial temporal receptive field. Originally, we have the ABCG sequence, so the cell experience the C goes to G. But when we change the second stimulus to G, from A to G, the cells has never experienced that. It's also a novel spatial temporal input to the receptive field. So there could be two theories for the temporal prediction. The cells are doing predicting the sensory input based on the history. For the spatial prediction, it's based on the instantaneous input. So to separate the two hypotheses, we designed this new experiment. We have the ABACG sequence. After training, we change the sequence to ABABG. If the mouse is really caring about the if neurons really learned the temporal sequence, we would expect that the response to the second B might be different from the first B. But if it's the spatial input pattern to the receptive field matters for this massive response, we would expect because B to G is will be a novel input. This transition will be a normal input to the V1 cells receptive field. We would expect the massive response would happen in this J period. This is how our result looks like. When we introduce a novel image, we still see a kind of a massive response as before, which means, yeah, this sequence works similarly as the previous ABCG sequence. And then we change the sequence to ABABG. So visually we can see the comparing to 2Bs, the response is pretty similar, but the response to the G actually is much higher than before, than originally the response to the G stimulus for both A and B cells. Yeah, so if you compare the response to the 2Bs, statistically it's not significant. If you compare to the response to the 2Gs, when we change it to ABABG, the response to the G will be higher than before, which is because B to G is a new transition and those neurons didn't learn before, because originally those neurons only see A goes to B, then A goes to C, and C goes to G, and B goes to G is novel to those cells. So we make this temporal duration interruption to go extreme. We tried to instead of remove one small set, we did that before. We included one extra small C with 3 Cs. We compared the response to the very last C with the response to the second C in the original ABCD trial. We found that maybe slightly the response became taller, but visually it's not very dramatic from this graph. But if we keep increasing the number of those Cs, here visually we can see that the response to those small Cs became brighter and brighter. If we do a quantification, we can see the response to the C cells respond to those C stimulus just keep rising. And also other cells, for example, D cells, they also keep increasing the response as we increase the duration. And we compared this with naive mice. In naive mice, the response seems flat, so it didn't keep increasing the response. So this kind of effect is not because of the artifacts of calcium dynamics. It could be the animal or V1 neurons since the time of the duration of the stimulus, and if it's very different from what those neurons learned before, it could increasing or accumulating an error signal. Yeah, this is pretty much all the experiments we include in the paper. Yeah, the behaviorally we didn't see any difference. And yeah, in summary, we saw increased neuronal activity to novel stimulus to complete green screen, and when we make the temporal duration interruption go very extreme, and the mouse will slow down to check out the image when it's novel or complete green screen, but not when the image is super long. Yeah, we have the bioarchive paper. Yeah, you're very welcome to read it and discuss with us. Yeah, so our conclusion is the visual cortex might work as those masked autoencoders to try to reconstruct the image learned before. And when there's a kind of big mismatch to the original learned the spatial patterns, there could be a big mismatch response, but not the time they seems those cells didn't doing the temporal sequence prediction. Yeah. Yeah, that's all it.
Thank you, Jinqing.
Yeah, thank you.
I have a sort of a mechanistic question, and I'm sure you've thought about it, because in the review we've tried to think about what are the underlying computational primitives that are implementing those predictions in the network, and so I'm curious what you're thinking, because you've established your task within this bond of parameters. There are predictions occurring, and so are you going to follow up with some specific experiments to uncover the mechanisms there? What's your thinking?
Yeah, good. Thank you for mentioning that. I read that kind of like a review paper, like our community review paper. It seems very rough and like all the details seems like has been touched. Yeah, I really enjoyed reading that very long paper. We did something kind of related, but so far we do not have a kind of a very I think we didn't reach to like a reliable conclusion. Previously, we recorded some inhibitory cells, and it seems we have the transgenic mice. We have the mice that label both excitatory and inhibitory cells. We have compared the difference between excitatory and inhibitory cells. It seems they behave like very similarly in those paradigms. I think one reason could be we didn't really separate the different inhibitory cell types, maybe like the PV cells, VIP cells, and they might function differently. I think in previous literature, there are like a positive error, say like neurons, and also negative error neurons. And if we do not separate them, and that could be the reason that we didn't see like different roles for different cell types. And for that's one thing we did. And also we didn't really get a layer 4 transgenic mice. So so far we don't know if this is because if this happens in the input layer or in the or mainly in layer 2, 3, or it relies on the feedback from anterior cingulate or prefrontal. Yeah, we do not know that either. Yeah, I think maybe you guys proposed some experiments that will touch those elements. So.
Oh, are you interested in running some of our design? I'll be
No. I just mentioned that we thought about that, and so far we seem like we do not have any conclusion about that.
Oh, okay.
I'll mention one thing, Ching, you did anesthetize the mice and see differences under anesthesia versus not under anesthesia. So that's one case that could be pointing somewhat to the mechanism.
Yeah, we did anesthetize the mouse. When we anesthetize the mouse, everything is gone. Even the visual response is weak, and also the big mismatch response was gone as well. So it seemed like, yeah, even it's no matter it's novel or familiar image, the response pretty much the same. I'm not sure if it's because different anesthetic or drugs. I think in auditory cortex, or I forgot, I remember when people anesthetize the animal, they still observe the audible response.
Yeah, but it depends.
Mm.
It's a part of discussion, but I think it depends on the design of the experiment. So, like for instance, if you have a really, really quick task where the sole mechanism of recurrence or like local adaptation is sufficient to make those predictions, then you might see a response. But when now you're in anesthetized mice and you have the whole network that is like in a weird stage, some of those mechanisms are not able to engage. So I feel like it depends on the task.
Yeah, I think those are pretty super fast experiment. I was a little bit worried if those super fast experiment actually relies on the physical properties of the cell, the memory potential, the very fast kind of some adaptation stuff. If those like a physical parameters that kind of related to the like an error, like a audible error signal. Yeah, I'm not sure about that, but in this case, yeah, we didn't see that.
Casper?
Yeah, at the beginning of the talk, Carson showed that most of the changes with exposure to these types of stimuli happen in the sort of adjacent, second order visual areas. Have you looked at these signals there?
Yeah, for the VR experiment, we also covered the medial area. Yeah, we have both V and medial. Yeah.
And your summary is that things are pretty similar across those areas. It's like the same story roughly, or are there any differences that you noticed? Forgive me if I missed something.
Yeah, that's true. For the VR experiment, it's pretty consistent for V1 and for medial and for lateral and even anterior region. But I think for the very long phase, I think it seems more dramatic in primary visual cortex. In medial areas, it's also statistically significant. One reason could be usually for the recording, I got more cells from primary visual cortex, which might make it be more significant than medial area. But yeah, we didn't see a difference, like at least in this paradigm.
Understood. Thanks.
I was curious if you, because the ramping behavior you have here is very similar to what some recent result from Farzaneh Najafi lab, where they show ramping behavior between stimuli. And so I'm curious if you looked at it in that light.
Yes, personally, I thought that they are different because I feel 2 years, 2 or 3 years ago, my first SFM poster is about ramping cells under the static image paradigm. It's the ramping cells when you present a static image without any training experience. There are, even with grading stimulus, naturally it seems like there are cells that they just ramp, and it's like the input is fixed, like it's just a one static input. You stay the image is just is staying there, and the response just a ramp. And here it's in this VR case, the input there, like even though you make the state stimulus very long, but those neurons just keep receiving like a different input as the mouse is running through this linear corridor. It's still changing inside the receptive field, even though it's duplicated. And we see this kind of keep increasing response. And first, this is a kind of population of like all the C cells average result. And we didn't see this effect in naive mouse. Yeah, I think for a static image, I did one experiment is in this kind of, in this ABACG paradigm. Previously, I trained the mouse to get a reward to the second A, and also did manipulations. This is a static image experiment. I made one image. I made the C image very long, and to see if there are any effect. In that case, I didn't see. Yeah, I didn't see any effect. I didn't see this kind of ramping behavior or big mismatch response. So I think it's this is the time sensing or this kind of error signal. It all still relies on you give continuous input to the visual system, rather than you just had you have a static image there. And yeah, I think that kind of ramping for some cells, they ramp to a specific image that could happen even without training.
Thank you.
Yeah.
Any other questions in the group?
Yeah.
I want to thank you both for presenting. We are at the hour. I want to emphasize that the data set we are planning to collect at the Institute as part of a community project is very interesting point of overlap with what you're showing. I think it might be helpful to your research program, because what we're planning to do is record with the mesoscope, with the NeuroPixel distributed probes, and diving deep into one cells with SLAP2. The same set of different paradigms compared against one another. And so I think it's going to be powerful data set to dive them in mechanistically. And then you could probably design experiment that are complementary, that just to dive deep into this. And so I'll be open to coordinate or discuss any potential overlap that will make the science better.
Thanks, Jerome. That's really helpful, and thanks for having us. I'm wondering if you've considered using natural images.
That's right. We thought a lot about. I've certainly thought a lot about this. The challenge I see is that, again, as you introduce new things, going to recruit different mechanisms. So the difference between gradings and natural image is that, like, the way you do pattern completion is going to be very different, and so you're going to see a certain recruitment of pathway that are coming from lateral and top-down inputs that are going to be involved now to make the prediction. And so I think we decided to do with gradings because these circuits are very well mapped. And so, like, we know what individual cell types are doing across so many studies. And so having this base, I think, is going to help propel the mechanistic effort. But if you guys want to do natural image in complementary, I think that would be amazing.
Thanks. Yeah, so I guess the other thing that's different is your experiments are all going to be single-day experiments. I guess that's the one other main difference. Or no?
No. So we have multiple cohorts. And so there's a cohort on the mesoscope that is going to be 8 consecutive days where we change the context. So it's 2 days at a time. And then the NeuroPixels is 4 consecutive recordings. Every day is a new context. And one of those contexts is actually sensory motor coupling. So I think everybody will get insight from this comparison.
Yeah, I think so. I think the one caveat with using the orientations is, like, I think the novelty responses could be different in V1, whether it's an orientation versus like a natural image. I think that would be the one thing that I would be most concerned about.
Yeah, as well, yeah.
That's right. So You might recruit when you're dealing with textures. Yeah, you might recruit a slightly different network, but I think that's the point. What we're trying to make is all of us paper are trying to test predictive processing, and they all have very important but little details differences that and then it's really hard to compare, right? Because then you had for instance, temporal predictions. It's probably the case that mice are very good at like very short-term predictions in V1, like we're talking about sub-second. But when you extend, then you need to recruit other areas. And so I feel like just these little things make it so that a study that I have like a 5 seconds window, another that has like a quarter second, you're dealing with recurrence on one hand, on the other hand, it's like hierarchical involvement of areas. So I think like these are really important.
Yeah.
Are you guys,
no, I don't. Yeah. Go ahead, Cheng. Sorry.
Yeah, I'm just curious. Are you guys also training the mouse doing a task, for example, giving them a reward in this
That's right.
paradigm?
That's right.
Mm.
They are trained to, some of them are trained to run on the wheels, some of them are trained to just see the sequence. But you've shown that does it matter?
I did like you mentioned from the Hofer's lab feedback, they said it matters when you give them reward or non-reward. Yeah, if it's only task engagement related, it probably does not matter that much. Yeah, I did. I don't know. I'm just curious if doing the task could change everything.
Well, so I think that's the choice, right? But I think the reason not to do the reward is because when you had reward to the behavior, now you're sort of engaging consciousness very deeply. There's like massive reward signaling that comes on top, and so that's going to modulate really strongly the response. Some people have shown that prediction error, as you said, just come up when there are rewards, and I think that's because the task is very hard, and so you have to rely on this boosting. In Sonia's overlap, they were 30 meters apart. Like it's very long.
Right.
And so that's why you need this gain mechanisms to push it up. That's my intuition. But then mice that work on a mice does not need a reward to map an environment. And I don't think that's necessary.
That's our perspective. Yeah. But
Right.
I think that having a closed loop is important, is what we think.
That's right. The closed loop is important. So that's why we had it in.
Cool.
Yeah, sounds great.
Great.
Yes.
Please feel free to explore. We share everything. So if you're more interested, we'll be happy to have your inputs.
Yeah, thank you. Thanks. And it's really cool how open this project is. It was fun looking through all your notes and stuff.
Thank you for presenting.
Thank you.
Thanks.
Bye.
Thanks so much. Bye, everybody.